Doctors Longo and Roberts discussed the current status of gene therapies in rare neuromuscular disorders in this 8-part podcast series. This is derived from the symposium that was presented at World Symposium 2025 in San Diego, California on February 4 through 7, 2025, and is intended for healthcare professionals only. This podcast includes information about investigational compounds that do not yet have a regulatory approval or authorization for a specific indication. The safety and efficacy of the agents under investigation have not been established, and contents of this podcast shall not be used in any manner to directly or indirectly promote or sell the product for unapproved uses. The views, thoughts, and opinions expressed in this presentation belong solely to the author and are subject to change without notice. The contents of this presentation do not constitute an endorsement of any product or indication by Astellas. In this part, Doctors Roberts and Longo will discuss treatment with gene therapies.
Can one administer AAV-mediated gene therapy repeatedly?
I think the traditional view would have been no. One can think of gene therapy as a silver bullet. Hopefully it will reach its target, but if it's not effective, that bullet has been shot, the immunological response has occurred, and it means redosing, at least with that particular vector, may become difficult. But this situation is changing and evolving as we have better understanding of immunological modulation for repeat testing. And we were discussing this yesterday evening, weren't we, Professor Longo?
Correct. So basically, the current AAV-based gene therapy cannot be re-administered. So if it is either effective or it doesn't work. And and the other thing is that even though in theory one could utilize a different AAV vector with different immunogenicity, there is many times cross-reactivity among the different adeno-associated viruses. Now, there are approaches in animal models in which you give a strong immune suppression to prevent the creation of the immune response against the adeno-associated virus, and at least in the animal model, it has been possible to give some of the gene therapy repeatedly. The second approach that is being tested is with gene correction therapy, in which, by using an RNA guide and the CRISPR-Cas9 system delivered by lipid nanoparticle, you basically correct some of the defective genetic information. Obviously, since this is done by lipid nanoparticles and not by an AAV, the immunity that you create is really not there. So you can give this one repeatedly, and in theory, it can be given more than one time. But again, you are absolutely correct. The current gene therapy cannot be given twice, and either it works or it doesn't work.
So will gene therapy-treated patients be able to go back to the standard of care or enzyme replacement therapy?
I think when we're talking to patients about the potential benefits of gene therapy and the amelioration of the requirement to have these infusions on a regular basis of ERT, the hope is that will work. But they need to be reassured that we can potentially go back to the ERT. So gene therapy is an important treatment, but we don't know the destination of the patient at the beginning, and we have to make it available to them to go back to ERT. One of the crucial questions, of course, though, is the basis of the immunological reaction that perhaps prevented the gene therapy being effective. So if it's against the viral vector, well, okay. If it's against the transgene, not great. If it's against the functional protein, that becomes more difficult. So it is somewhat, I think, at this time, to be fair to saying to patients, think of gene therapy as a trial treatment. It is somewhat a leap of faith and an important observation, of course, for the patient community, but just be aware there may be downsides.
And I totally agree with Dr. Roberts. In general, they should be able to go back to enzyme replacement therapy if the gene therapy is not effective. However, what we are starting to appreciate is that we need to understand the immune response, not just to the enzyme replacement therapy, but also to gene therapy. So it is what this field is doing is forcing geneticists to deal with the immune response, a field that historically has not been dealt together. So the two things need to be integrated. The advantage of the gene therapy is that the protein is produced endogenously. So there should be the development of some degree of tolerance with time in the body toward the endogenous production, endogenous continuous production of a protein. Now, will that happen all the time? I still do not know. Again, we need to understand much better what is the integration of the immune system with the response to gene therapy in the ongoing clinical trials.
What are the potential factors that impact durability of gene therapy?
So if one takes the in vivo approach, where, of course, we're seeking to have the episome producing the DNA there within the cell, there is the possibility that that episome may not be retained, and that could impact the durability of the response. By contrast, the in vitro response, where, of course, one's hoping very much for inoculation, if you like, of the host genome, may be a more durable response, but is far more intrusive as a treatment approach. Effectively you're giving an autologous hemopoietic cell transplant.
We already have evidence of the ex vivo gene therapy that it is of long duration and remains effective for quite some time, in a way that it is comparable for many aspects to a standard bone marrow transplant. We know much less about the AAV. So one of the concerns that is around is the fact that if you deliver AAV therapy to young children where organs are still developing and multiplying, there is the possible loss of the gene correction in daughter cells that are generated obviously by cell division. We still do not know enough about it, but it is a theoretical possibility that still it is in our mind. So should we be doing gene therapy in organs that divide very rapidly in children, or should we limit our trial to adults? For now, many of the trials you have seen have been done in adults, but some of them have been done in children. However, in the one done in children, many times they affect the central nervous system, that it is not an organ that divides very rapidly.
Could you clarify for patients in gene therapy clinical trials who've discontinued ERT over a long time, what was that transition like? Did patients stay on enzyme replacement therapy while in the trial and then taper off, or another approach?
So basically, when do we choose to discontinue enzyme replacement therapy in patients who have received gene therapy? So in my own experience, basically there were data emerging from the biological testing. So what you do, many times you measure the activity of the enzyme either in serum or in the cell, or you use additional biomarkers that usually are utilized to track progression of the disease. When you have a consistent reduction with maintenance of body function, in the clinical trial that I did, there was maintenance of the respiratory function and the 6-minute walk test. You stop the enzyme replacement therapy, and then you keep monitoring all of the same biomarkers, with the idea that if you have a worsening, then you would go back on enzyme replacement therapy. Obviously, there was no worsening. The patient was feeling happy and didn't have to go back to clinic every 2 weeks to do the infusion. And there was actually an increase in physiological function of the patient. So the patient decided not to go back to ERT, and they stay there. Now, will that happen that way all the time? I do not know. But at the same time, I think that if you have good biomarkers, good endpoint that you can assess to determine the functional effect of the gene therapy, you reassure that whether something is effective or not effective.
Can you comment on the use of non-viral vectors for lysosomal storage diseases, especially after the technologies developed for the COVID vaccines?
I think this is a very exciting area, and we've already alluded to the fact that many intravenously delivered viral vectors will not get through into the central nervous system. Nanotechnology and new nanoparticles offer the possibility of delivery into the CNS without the need for invasive technologies, for example, directly into the ventricular system. So I personally think that's going to be an extremely important step, because if one thinks, for example, about the model of Pompe disease, and as we've all seen, the huge amelioration of the natural history with enzyme replacement therapy, we expect with gene therapy we'll see a similar issue But we have to think about the CNS involvement and those children who are coming through into our practice with learning needs. So a nanotechnology might be helpful, or two different gene therapies, one to target CNS involvement and one more peripheral neuromuscular involvement, might be something that really leads us closer to the amelioration, not a cure of the disease, but closer to the amelioration of the condition in the longer term and not just in the short term.